Is the whole committee here? Eric, is it, or Lisa?
I do not see everybody yet.
Okay, we can
I think
wait
I
a
see
minute.
everybody actually.
Yes, everybody's on. Sorry.
Yeah,
Okay,
just a lot of names.
great. And you're ready to go, Eric?
As much as I can, yeah.
Okay,
Oh yeah,
good.
I have to.
Great. Well, welcome everyone to our D-Mice Informatics Conference. And as you know, we have a lot of extra conferences at the end of the term, people defending theses, dissertations, proposals, etc. So today we're going to have a master's thesis defense by Eric Leung. And many of us around the department know Eric. He's been around for a while, and he is finishing up his master's degree. Just for those of you who are not regulars at our conference, Eric will give his presentation, and then then we'll be open to questions from the public. The public will be excused, leaving behind the committee and Eric. They may have some further questions for him. Then he'll be excused, and they'll make their decision on whether he successfully defended his thesis. So just by way of bio, Eric is a Master of Science student in the bioinformatics and computational biomedicine major. He got his undergraduate degree from the University of Oklahoma, where he received dual bachelor's degrees in math and biochemistry. He leveraged his analytical and biological backgrounds to pivot into biomedical informatics and study the human microbiome. He's also often involved in community efforts in open source software and open science. So he will defend his master's thesis today. All yours, Eric.
Cool. Awesome. Thanks, Bill. Share my screen. All right. Move stuff around. Oops, I guess I moved the slide. All right. So I'm assuming you can see the slide. Let me know if I can't. This is my title. So today I'll be talking about, the research I've been doing. And to sum it up, it is talking about the urinary and vaginal microbiome community structure in women with urgency urinary incontinence. And I think somebody's mic is on. Somebody could mute that. Okay. So the title was a bit of a mouthful, so I'm going to take a little bit at the beginning to talk about the three parts of that. So what is urgency urinary incontinence and why should we care about it? And then how the urinary and vaginal microbiomes may play a role in this condition. And then transition into looking at the community structure using these co-occurrent networks using more network-based methods. I'll then go into my aims of my work, which are split into more of the traditional way in which we analyze these methods, and then exploring more of these network-based ways. Without further ado, so I've spoken a lot about urgency urinary incontinence. So what is it? So urgency urinary incontinence, or UUI, is the involuntary leakage of urine that is preceded by a strong sensation to urinate. And so this strong sensation is this urgency part. And so this is difficult to defer for individuals with this. And so this condition does affect both men and women, but by surveys and estimates, it does affect more women than men. And additionally, other conditions that are posed as risk factors for UUI are increased age and increased body mass index. Moreover, this condition has a high economic burden, both from a direct medical cost and from indirect costs from loss of productivity. As you can imagine, having to have a sudden urge occasionally and randomly. And additionally, this condition has detrimental effects on, say, a person's quality of life, For example, an individual may have embarrassment among their peer group, having to leak, or even having to plan out routes to the bathroom when they might have the urge to void. Now, there are treatments for urgency urinary incontinence. Namely, there are these three broad categories that are typically described, which increase in invasiveness as the previous treatment becomes unsatisfactory to the patient. Now, the least and most invasive procedures are the behavioral techniques, such as time voids, and then more surgeries and invasive techniques, injections or surgeries. Now, the largest class of treatments, which I'll focus on, are these medications. And typical medications that are used are anticholinergic drugs that are designed to relax the muscle around the bladder to prevent you from having to go so much. Now, focusing on these medications, they do come with a range of side effects, such as dry mouth, dizziness, constipation, and blurred vision. Unfortunately, to make things worse, these medications are ineffective for a very large population of individuals with UUI who take this. So this suggests that there are etiologies outside of targeting the neuromuscular function of the bladder to treat this condition. And so stepping back a little bit from the bladder, one large piece of variation that contributes to urogenital health and disease are microorganisms. And so urogenital here will involve the reproductive organs, including the vagina, and the urinary system, including the bladder. And so there are a few bacterial infections that affect the urogenital area, such as bacterial vaginosis and sexually transmitted diseases and urinary tract infections. And now, considering the impact of these microorganisms on the urogenital health, we should consider the impact of these microbes with urgency urinary incontinence. And so going back to the bladder, so we have this strong role of these microbes, but going back to the bladder, we can see that medications, again, will aim to relax, say, the bladder lining here. And with recent studies, or not recent studies rather, the bladder is not just a passive barrier made up of urothelial cells. Rather, it can react to stimuli within the bladder. And so much attention has been concentrated on the role of urothelial inflammation and infection in the urine that may cause sensitive bladder lining to unnecessarily trigger the bladder that can lead to urgency urinary incontinence. Now, historically, we've thought about the bladder as sterile. However, this paradigm has been challenged in the past decade. And so for context, here's a standard urine culture after 24 hours on a blood agar plate. And so there's no growth here, there's nothing growing. However, with new expanded quantitative urine culturing techniques, using this slightly more urine, so instead of 1 microliter from previous, we're using a lot more and growing it for longer, there is growth from the urine. So one possibility here is that UUI symptoms are influenced by these microbes that inhabit the bladder. And sure enough, studies have found changes in the microbiome, or the bacteria in the bladder, or the urinary microbiome, that are associated with UUI. And here I'm just showing a subset of studies studying various changes in the urinary microbiome with respect to UUI. And so I brought up earlier that the microbiome is important to urogenital health, and the changes in the urinary microbiome are now associated with UUI. Now the question is, how does the vaginal microbiome change with the UUI? And so this is still an emerging work to understand this possible relationship and how might we look at these two together. And also the fact that the vaginal microbiome is quite well studied and is a large reservoir for bacteria, especially the large dominance of the lactobacillus bacteria as well. Now I've talked about how these microbiomes and their changes and making inferences about that. And so what I mean by how do we quantify this? And so a very simple example here is here I have two circles indicating maybe two samples we're curious about with the different species as these boxes that we're interested in. So typical questions we might want to ask is the number and distribution of bacteria. And we can quantify that as alpha diversity. And so this will look at the, I mentioned the number and distribution of these bacteria. And so you can create replicates of these and versus different groups and compare them. And similarly, you can also take each individual sample and then compare the different diversities across between each other and create an ordination plot here so you can more spatially understand these. And so what's missing from this is that, or rather, these would be what I would call more common or traditional methods of how we would study these microbiomes. However, these methods are, they make assumptions about just studying these bacteria in isolation. And one title actually summarized this very aptly, that diversity is just a question and not the answer, because we see this diversity and then you might say, so what? And then what populate or created that certain changes in diversity? So we must look a little bit deeper to understand what may give this diversity. And so what's missing here is the organisms will actually coexist and have potential interactions with each other. So here on the right, I show a table of these interactions, or their ecological interactions rather, between pairs of organisms. And so there are pairs, there are more positive interactions, such as cooperations, where both organisms benefit. And then there's more negative interactions, where both organisms may lose by, say, competing for resources. And so this is just a pair of organisms here, or rather in bacteria in my case. However, we can scale up this table of interactions to a community to abstract, say, this invisible network of interactions. And so here is a such diagram where the black circles here would represent each of the bacteria, and these edges or lines between these may represent the different interactions from this table. And so these missing interactions can be, from our analyses, can motivate these more network-based methods that are emerging in their use in the microbiome world. And so with that, I'll get into my aims. So first, I'll characterize the microbiome of women with and without UUI using these more traditional methods for both the urinary microbiome and vaginal microbiome separately. And then I'll do a very similar analysis, but then using more network-based methods to then characterize each of the urinary microbiome and vaginal microbiome. So I'm going to go in the next few slides to talk about the study design we had. So first, this was part of a larger cohort study that we had. And in the study here, I looked at 50 women, 30 women without urgency urinary incontinence, and 20 women with UUI. There were a number of exclusion criteria based on some factors that may be confounding based on our study. So some include needed intermittent catheterization or known neurological disease that may affect bladder function. And individuals included in as UUI are predominant or urge predominant incontinence confirmed on a 3-day bladder diary. So some of the information that was collected was demographic information, such as age and BMI, this 3-day bladder diary that I mentioned, which is a bladder diary that they take. 3 days, they account for what they've drank and their voiding patterns and how they felt. And then clinically validated surveys on symptoms to quantify how they felt and their severity of these symptoms. And of course, we collected urinary and vaginal samples to then analyze that and understand its relationship with UUI. So here's a brief summary table of what we found between our UUI and control group for their demographics. So here, notably, their age, BMI, and history of recurrent urinary tract infections were significantly different between our two groups. And other notable demographics that didn't differ between these are menopause status, estrogen use, and race and ethnicity. A few of the questionnaires here, these are more the symptom questionnaires we asked our participants to take. And so here, here are 4 of them, and all 4 of them were significantly different between the two. And this is expected considering UUI patients have this strong urge to go. And so takeaways from just the study population here is that we see that individuals with UUI are much older and have higher BMI, something that is a known risk factor that we found. And then we found that individuals with UUI had significant urinary tract symptoms, also expected. And then menopause status and estrogen use were similar, and that was based on how the study was designed. Okay, so now in the next few slides, I'll take you through how the samples were collected, processed, and then prepared for final data analysis to then get into understanding the microbiome. So previously in my background, I mentioned that bacteria in urine was determined using culturing techniques. However, there are now more high throughput ways to understand these by targeting the 16S gene that exists only in bacteria. And so we can target that to then study the microbiome. And so first we took catheterized urine samples and vaginal swabs. Both were taken by a trained clinical professional, and we then targeted and extracted the bacterial DNA, and then amplified those sequences to be sequenced. And after we get those sequencing reads back, then we process those sequences using data2. We remove chimeric sequences, and then assigned our taxonomy using the Silva database to the genus level. And then we built a phylogenetic tree for downstream analyses. Okay, and then one more prep side. So here are 3 large steps that we took to then prepare this data to be analyzed. And so first, did some pre-processing to remove out some lowly abundant bacteria that may not expect or maybe from noise. Then with such low biomass such as the urine, we took some precautions and removed contaminants using the decontam algorithm. And then we did some final processing to then keep expected in there from the literature and then remove known contaminants before further going into data analysis. Okay. So now we're at the data ready section, and then now here I'm going to go over some of the traditional analyses that I'll show a subset of. And so here are a brief number of more of these traditional analyses. So first we can do a unsupervised or clustering approach to understand how generally the compositions of these bacteria associate with clinical information. And then we can get into these more diversity measures that I briefly alluded to at the beginning. So looking at the number and distribution of bacteria between groups, and then how those diversity between the samples may change between each other and we can more visually see how each of those samples are different. And then another piece we could do is what's called differential abundance, to look at individual bacteria and how they may differ between groups. So firstly, again, I'm going to start with this unsupervised, or this clustering approach, to see what composition of the bacteria may relate to our clinical observations and cohort status. So the data here were clustered using relative abundance. And so here I've shown a stacked bar plot of that. So here the x-axis is our samples, the y-axis is our relative abundance, each of our genera, and each of them are colored by their top genera here. And so we performed hierarchical clustering on our samples using the weighted unifrac distance, and found two groups based on the silhouette metric to choose two groups here. And then these two groups by our microbiome data do not appear to associate with our cohort status or having UUI or not. And then exploring further these clinical relationships, we see that there's no relationship between vaginal product use or estrogen use. However, there does appear to be an association in these groups of menopause status and relative abundance of lactobacillus in the urinary microbiome here. Now we can do a similar analysis to our vaginal microbiome. So very similar setup. Here, stacked bar plot. X-axis is all the different samples from these same women. Relative abundance. The colors here match the same colors as before. We perform hierarchical clustering. Did the same clustering and using the silhouette distance or metric to identify their two groups. I also found that there's no grouping between these cohort groups, or rather the hierarchical clustering groups and our cohort status. And then looking at more of these clinical pieces of information, so we don't see a significant association with menopause status or estrogen use between these two groups. However, there does appear to be significant use of, or rather a significant difference in use of vaginal products and the relative abundance of lactobacillus in these two groups. Now we've gone through a little bit of the general unclustered, or rather clustering between samples. And here we can look at now the distribution and number of bacteria in each of them. So here I'm going to focus on just the urinary microbiome. And so one quantity, or one measure rather, of diversity, alpha diversity, is observed number of taxa. And so here I have between our UUI group and controls, and then after controlling for age, BMI, and menopause estrogen status, we see that there is no difference between the alpha diversity of the urinary microbiome between UUI and controls. And furthermore, here are two other common measures of alpha diversity, the inverse Simpson and the Piley evenness. Also adjusting for the same covariates, we see that there is no change in the urinary microbiome alpha diversity with UUI. And similarly, we can apply these same measures to the vaginal microbiome, and consequently, we don't see a difference in the alpha diversity of the vaginal microbiome in UUI and controls as well. So this suggests that the number and distribution of bacteria may not change with having UUI. And another look at how these diversity changes between samples, we can look at the what's called the beta diversity here, and find that they also don't vary with UUI either. And so on the left here, I'm showing a ordination plot using our urinary microbiome samples. And after performing a per MANOVA test to assess whether our samples grouped by UUI, we find that no relationship between how the diversity changes between these samples and having UUI. And similarly, as you may guess, I'll do this same analysis on our vaginal samples here, and we see a very similar result that the vaginal microbiome doesn't associate with UUI. And so if it did, you might see certain colors here the colors here represent the having UUI and controls. And if they were associated with UUI, then you would see all the maybe salmon colored in one corner and all the like more teal or blue in a different color. So you see here, they're all kind of spread apart. So as a takeaway from these more traditional methods, so based on the urinary microbiome, we don't see any difference between the different cohorts based on our groupings. However, we did see some effect of having different menopause status and affecting how the urinary microbiome varies. And then for the vaginal microbiome, we also didn't see a change in the associated with UUI or being in control. However, we did see that having some vaginal product use actually does influence the variation in the vaginal microbiome to cluster samples in a certain way. And then in both, we found that lactobacillus is quite a dominant feature in influencing this variation. And then the last couple slides show that we didn't find any difference in the alpha diversity or the distribution and number of bacteria, or the changes in those diversity values. So at the beginning, I mentioned these possible limitations of these traditional analyses, not taking account of these, the context of the bacteria and what they might be doing. And so here again is this table of ecological interactions that I brought up previously. And so using these more network-based methods, we can try to abstract these interactions up to a community level and generate a hypothesis, if you will, of the interactions that may occur in our community. And so this might be one such way. And so going forward, so again, the circles here will be the different bacteria, and the edges will be some sort of relationship that we may define. And so because we don't have the ground truth, we'll have to abstract or have a proxy for that as a certain edge. And so what kind of edge or relationship will we define here? And so the relationship that I'm going to define here is a correlation. And so to describe what that may look like, so here if I focus on just bacteria A and B here, and then we look at how the abundances of them occur in different samples. So here, these different orange circles are the different samples that we have. And as you can see, as say B abundance goes up in one sample, or as it goes up, then we can see that A correspondingly also goes up. And so here we could say that there's a strong correlation between them and may suggest some sort of interaction that is occurring between them. And so this is mapping it back to these ecological interactions. And then another point I want to make is, what if there's no edge between these two? And so that could also occur whereby, say we have this other bacteria C, where the abundance of this changes across different samples. When, say, looking at A, it doesn't change at all. And so here it would have a very weak correlation or no correlation at all. So you wouldn't have an edge there to connect them, suggesting any interaction. And then to keep the things a little bit simpler here, I'm just going to be focusing on just the positive correlations for the network going forward. So that's a little bit of background about all these networks. And so here's a very brief pipeline of how to take the microbiome data that we had previously and then transform it into a network. And so we take the abundance matrix and we then calculate these correlations. And I use the sparse CC correlation algorithm, which is designed for microbiome data. And then we performed a permutation analysis to then generate a null correlation distribution to then threshold correlation set to remove any more spurious or lower correlations. Then performed more of these network measures and module detection algorithms to then make use of this now, I guess, our putative network structure, community structure, and identify properties of it that we wouldn't have otherwise without the network. And then we can visualize and then make more inferences about what this network may mean. Okay, so in the next ending part here, I'm going to spend time on talking about the urinary microbiome, and then I'll switch gears and then talk about the vaginal microbiome networks that we have. So starting at the urinary microbiome, so here taking the same samples we looked at previously with the traditional methods, I constructed a network for our UUI and then one for our controls. And then see here, I've colored them based on a clustering algorithm called InfoMap. Sorry. And then here, InfoMap identified it allows us to try to identify some structure in this based on how the network is constructed. And so with this algorithm, we've identified there are 4 groups in our UUI network and then 9 groups in our controls. And a little bit more about these networks from a high network level. So in our control network, we appear to have a lot more bacteria. So we have 135 bacteria in our control network versus 93 in our UUI. There's also subsequently more edges or associations of how bacteria co-occur in our control network. And then this modularity measure, it quantifies how well the network will group together. And so for a control group, we see that it has a higher modularity measure, which means that it, at least by this InfoMap algorithm, it does group them a little bit better than, say, the for our UUI group, this algorithm. And then our normalized connectance measure quantifies the number of observed edges that we've had, or the co-occurrences we had out of all the possible co-occurrences we could have across all of the bacteria in them. And so it appears that there are more co-occurrences in our UUI network, which may suggest more variation in how many unique different bacteria may co-occur in our UUI network than in our controls. So this is great at a very high network level, which is what we wanted to look at, which is the community structure. And then now, how about we get into some of the actual identities of these bacteria? And so here I've taken that same network from before, but then annotated it with a phylum level. And so here, as a lot of these network analyses look like, they kind of look like a hairball. So I've taken some time to redraw this to make it a little bit easier to focus in on the phylum level and seeing if there's any association differences between them. And so what I'm going to show you is what's called a hive plot. And so on the top right here is a representation of what looks before. So I'm taking most of the data from here and then just mapping it onto this, what looks to be a star diagram for now. And so I've taken all the different phylum and aligned them onto the different axes. And so say this vertical one right here, this more salmon color is all the actinobacteria in our, at least UUI group. And then this purple would be all of the proteobacteria. And so here we can more easily align and push and focus on each of the different phylum. And then I then take all the edges and then I just draw them between the corresponding bacteria as they align across this axis. And so this may look something like this. And to make it easier to visualize, I've only focused on a little bit more stronger correlation. So at 0.5. So it'll look like this. So again, to interpret this is, say for this arc up here, so it's connecting an actinobacteria here to a Bacteroides. And so this would be one correlation between them. And so forth, you can make similar associations with the other edges. And so this is for our UUI network. And if we redraw our controls, we get something like this. And so some things that are notable is if you notice, say for firmicutes here, we see a very dense cluster of edges that resonate from around here. And there's a lot more that are going from the firmicutes here to proteobacteria that are not as prevalent or prominent in our controls over here. And so looking at not only just those two, I did look at the pairs of phylum and compared it between the groups. And there does appear to be a statistical difference in how the different phyla may associate in UUI compared to our control group, which is very interesting to see. Now, so that's at the phylum level. So let's look at more of the genera level. And so here I want to look at, well, one question is what may be important to the community. And so again, these network-based methods allow us to abstract or create a hypothesis of what the community is doing. And so with this network structure, we can identify important bacteria in our community. And so one measure of that is what's called betweenness centrality. And so here's a very simple toy example of what that may look like. So this blue bacteria will have a high betweenness centrality, which loosely speaking is because it's between most of the other nodes or bacteria in our network. And so I've calculated the top betweenness centrality measures for each of our UUI and control network, and then show them here. And so one thing that we notice is that lactobacillus is actually the most central based on our betweenness centrality in both of these cases for urinary microbiome here. And so one thing you may be wondering is what may be the different connections outside that are connected to this bacteria, even though they're both central to each of these. And so here I want to look at that overlap. So here, again, I'm focusing on lactobacillus because it was found central to the urinary microbiome in both of these networks. And I'm taking a look at the surrounding bacteria in each, and then comparing the overlap of those bacteria. And so what results is this Venn diagram that shows that overlap. And so we can see there are some differences and some similarities. So there are, say, 10 of these bacteria that are common between our UUI and controls. They're both associated or correlated with lactobacillus. Then we see 16 that are unique in our controls, and then 25 bacteria that are uniquely associated with lactobacillus in our UUI group. So I want to take a moment to take a look at what these 25, at least what some of them are, real quickly. And so some notable, so looking into some of these here, we see that a lot of them are associated with some sort of urinary tract infection. So some of them are more well-known, like E. coli here at the bottom, and the other ones here are emerging uropathogens as well. So this is very interesting to see that lactobacillus can be a very strong player, but also be associated with these more uropathogen players as well. So just a brief summary of the urinary microbiome network findings here. And so based on just strictly network measures here, that the UUI network here is less clustered, or they less in these structured groups than our controls. The UUI network has more observed microbial co-occurrences, or more of these edges between the bacteria. We found that lactobacillus is central to both of these, and that when we look at the UUI group that are specific and associated with lactobacillus, we say that a lot of them are associated with some sort of infection. Now, switching gears slightly, we're going to go into the vaginal microbiome now. So here is a visual representation of that, just as a quick visual. So again, same clustering algorithm to just identify just general structure here. So we have 5 groups in our UUI group, and then 6 groups in our controls. So just briefly looking at some of the statistics here, the number of bacteria in each of these is actually very similar. However, the number of edges, or the number of associations, co-occurrences we see for our UUI group is a lot more than our controls, being 396 to 195. And then based on the structure of these networks, the modularity of how the clustering algorithm worked, the modularity appears to be the same. And then normalized connectance here, because it's, again, as a reminder, it's related to the number of edges that we see versus all possible ones. And because there's a lot more edges here, we can see that there's an increased normalized connectance here. Okay, now very similar to before, I want to focus on the phylum level information, but again, here is very difficult to see, so I've remapped everything around to look like a hive plot. And so here again is what that would look like for the vaginal microbiome using the networks correspondingly. And then so here we can also see some slight differences in how there are a lot more associations between, say, Bacteroides and Firmicutes in our UUI versus in our controls, where we don't see as dense of associations or other stronger associations between them, and so forth. And then similarly, I also compared all these different pairwise correlations of the, or not pairwise correlations, taking all the pairwise like number of associations between these different phylum and compared those, and there does seem to be a difference in how the phylum associate in our UUI compared to our controls. And then going in a little bit into the bacteria themselves, or the individual nodes, again, I'm looking at the centrality measure, or what's most central bacteria in these networks for the vaginal microbiome. And so for our controls, we see that lactobacillus is quite central to this network, which is unsurprising considering its dominance in and importance in the vaginal microbiome. However, for UUI cases, we see that we have this other one called Aerococcus as being the most central or important one to this network. And it's in between this is much higher than the rest of them. So I'd like to dig into it just slightly here. And so actually, there's been some recent research on this bacteria, and one species of Aerococcus here is actually associated with UTIs, urinary tract infections, in the urinary microbiome, actually published earlier this year. And so how is this related to talk about the vaginal microbiome? Because again, this Aerococcus was found in the vaginal microbiome here, or rather, most central. So I mentioned that we're interested in the vaginal microbiome because of its close proximity and serves as a potential reservoir for infecting bacteria, such as Aerococcus and some Aerococcus or other urinary tract infections here. And then furthermore, to push or to to add to the story of the potential infections, here I'm looking at focusing at the Aerococcus in our UUI network and looking at the neighbors or whatever else it co-occurs with. And here at the top two here that I have based on average relative abundance, we have Gardnerella and Prevotella. And looking at the literature here, these two are actually quite prevalent in being associated with a urogenital infection called bacterial vaginosis. Again, another type of infection that may find its way through the urinary tract to then cause possible issues with UUI. This is some summaries on the vaginal microbiome here. So again, based on our large network measures, the UUI group is less clustered based on these large topological measures, and there are also more observed co-occurrences of these bacteria in our UUI group. Then we also found this Aerococcus genus as being central to the UUI group that wasn't found in our controls. And furthermore, when we looked at Aerococcus, it is can be found in the bacteria that are correlated with Aerococcus. When we look at the bacteria in our UUI group, those bacteria are commonly found with vaginal infections. And then, so to wrap up all of the analyses we've seen, so way back, we talked about more of these traditional analyses, and we didn't find any microbial differences in both the urine and vagina between our groups UUI and our controls. And so this may suggest that the microbiome analyses, or the traditional ones rather, don't account for these ecological interactions that may be important to understand these more nuanced differences. And so here I've shown that network methods may help in exploring this abstract and invisible microbial community structure that we don't have immediate access to. And then using that, we can then identify central bacteria in these structures and find that some of them may be associated with urogenital infections. And then going forward, this may play a role in devising more hypotheses on how we might treat UUI going forward. And so with that, I would like to thank everybody who's helped me along the way. Very importantly, my committee, Lisa, Elish, Guangming, Tim, and Mark, who isn't unfortunately passed away last year. But yeah, thank you to everybody. It taught me a lot along the way. I hope to take all your teachings with me going forward. The Division of Urogynecology for all your clinical expertise to help me understand UUI from many different perspectives. Thank you for your time and attention as well. The Carson's Lab for being a home for me to grow and to share my work, and also to listen to everybody else's very cool work. The Rosenbaum Asquith Lab for helping me with the sample processing and also being a great group to hang out with. The DMI staff and fellows for all their support and guidance along these years, helping me get through this. Friends and family, and then of course my funding sources, the SUFU organization, the NLM, and then IDDK. So with that, I will take questions. Thank you.
Great, thank you, Eric. Any questions?
Yeah, thank you.
I'm sorry, go ahead. Someone.
That might be
You can either unmute or type into the chat box. I guess maybe one Well, one thing that's interesting, and there's a number of clinicians, and I've chatted
Sure.
to Lisa about this in the past. The teaching in medicine, at least when I was in medical school a while ago, is that the urine is sterile,
Yeah.
and only when there are infections can we grow out bacteria. But clearly, the microbiome is there, perhaps not detectable by the normal clinical methods, that physicians have for diagnosing things like urinary tract infection.
Hmm.
It's just, and then maybe I guess a question. So, I mean, clearly there are differences here. And perhaps this is beyond the scope of your work, but are these differences? What's the cause and effect here? Um, is
Hmm.
the microbiome causing these conditions, or are these conditions somehow causing the changes in the microbiome, which I recognize may not be answerable from the work you've done.
Sure. Yeah. As you suggest, yeah. I don't know if it can be answerable. It's a fascinating question. I think it leads to a lot of different um, possible, grants that could be written, right? But yeah, I mean, one thing I bring up here is like the context of the vaginal microbiome, and so I think that has a really nice avenue to explore. One idea is to kind of leverage this more ecological thought. Somebody gave I think it was a conference. Somebody gave me an idea um, to try to simulate how the vaginal microbiome hypothesis. If your hypothesis is that like the vaginal microbiome like completely seeds the urinary microbiome in its former quote unquote sterile state and may seed it more. You could do like some sampling to see if you can reproduce or like how, sample from the vaginal microbiome into like a simulated urinary microbiome and maybe compare them. And that could be a way to understand at least the creation of the urinary microbiome. But then I think your question also talked about the more disease state and like which one is like a cause and effect. Is that right?
Hmm.
Um, yeah, that's a hard problem, and I think with the microbiome, like even working on this, it's very hard not to think about all the different confounding factors that you have to consider when doing like very causal microbiome research, considering like microbiome or like bacteria or they exist and uh, developed between individual like between other bacteria, but also shaped by their environment. So yeah, that was a long-winded answer to say yeah. I'm not sure either. So
Yeah, no, that's fine. And there's some, I don't know if you can see it because it's hard to see the chat box when you're screen sharing, but there's a question from Rose and another question from Mina. So,
yeah, okay, yeah, I can see that. So yeah,
okay.
Rose, do you apply your machine learning analysis directly to any other microbiomes, or would you have to tweak your processes per microbiome? Oh yeah, that's an interesting thought. I mean the methods are so the network part of it could be applied to others, but I feel like because the microbiome is very heterogeneous, it there's a lot of nuances that I've come up with along the way that make it more difficult to analyze. Like for example, at the very beginning, I showed that there's a lot of um, these. I guess I could show it. At the very beginning, I did the like clustering analysis and showed that there's quite a bit of structure in these like two groups. Where is it? Yeah. So like here, there's like some major groupings in our microbiome, and this isn't surprising. Other studies also will try to group and identify like these certain sample or participants with very dominant bacteria, especially in the urine and vagina. And so another long-winded answer to yes, you would have to tweak it to apply to other microbiomes. And I think that's what makes microbiome research very difficult because of this like variability. So, oh yes, and then Mina, thanks Mina. Let's see, negative correlation will be interesting to explore. Differences in unique bacterial colonies between UUI and UU, or controls and UUI may be due to some negative effects. So I guess more of a comment on negative correlations. Yeah, so I mean, yeah, negative correlations, my committee also asked me this question, and it's an interesting area. The more, it seems very intuitive, and the more I looked into the literature, people were very uncertain on how to make use of it. And because the negative correlation, so a positive correlation, sometimes I've used the term co-occurrence, or things that co-occur or you see them present together. But doing the opposite, where like one bacteria goes up and another bacteria goes down, is very difficult to ensure or say. So one analogy somebody helped me develop, or like, just talking it out, is kind of like being at a bar. That like if I keep on going to the same bar, but like you aren't at the bar, does that mean we necessarily don't like each other or have some beef? Not necessarily. And then also there is actually some specific work. I saw some publications that came out earlier this year that actually designed specifically to look at negative correlations, because when you look at a bacteria as this example of having like all of one bacteria and then seeing none of another, the shape of your data and how it looks is very difficult to infer. So that'll be interesting. Yeah, so long story short, my answer to that would be, or comment, is that looking at negative correlations in the microbiome and also how to incorporate with the positive correlations is an open area to explore, because yeah, it's difficult to assess what that might mean and how to accurately quantify it. So thank you. All right, Jennifer, I see your question. Let's see. What do you see as next steps for this research? What needs to be done before treatments can be developed for UUI? Ooh. Okay, so first off, I'm not a clinician. Second, this is a great question. I think, let's see. Do you see next steps? So it's difficult for me to answer because I've added the, so the vaginal microbiome is new, so that adds a whole other piece of information there. Let's see, treatments can be developed. Yeah, so hypothetically, maybe the end product would be some sort of probiotic or bacterial supplement or cream that could be applied with more evidence and support on having the vaginal as being a place for influencing the health of the urogenital tract or urinary tract in general. I know there is evidence to show that, if I recall correctly, using like topical estrogen cream can reduce the number of recurrent UTIs. So one thing could be possible is developing, an end product may be some sort of probiotic cream that could be applied to change the vaginal microbiome, to then maybe help the health of the bladder microbiome. And so to get there, you would then have to identify how changes in the vaginal microbiome could create or be associated with a healthy urinary microbiome. So I'm kind of thinking backwards from like the treatment back to how you do it. So I guess what would be next is how to just get more samples or funding for looking at specifically like more vaginal and urinary microbiome samples that are collected together, so you can see how both of them vary, because I don't think it is very feasible to directly influence the urinary microbiome through, say, an injection or something. So, okay, let's see.
Great.
Yeah, I know we're at time, so let me see what other. Okay.
Okay.
that
Well,
addresses it.
thank
Yeah.
you, Eric, for a great talk. And what we'll do now is we will dismiss the general audience, including myself, and leave you to your committee who may have further questions.